currently at SWIMS!
We are here to learn more about coral reefs from Dr. Phil Thompson. Moments later, we'll go in and know more about different species of coral reefs and think of solutions to protect them.
First of all, welcome to SWIMS, everybody. I'm Phil. I've been working here since about 2014, and I got my PhD in studying the physiology of corals in places like Hong Kong, where we have these big seasonal temperature shifts. But what I thought we would do, since you don't have a lot of time here, I don't want to spend too much time just talking. I'd like you guys to come up here and observe a few things. This collection represents maybe about 15 to 20% of that. The genus, pretty much everywhere throughout the tropics of the Pacific Ocean, we have 4 to 6 species here. And the reason I say it's a bit of a range is because we still don't know what species are what, because it seems that there's been a lot of genetic hybridization in this part of the world. So a very common one that we find around this area in Hong Kong is called Solitariensis. This is a coral that it's actually on the IUCN Red List. It's a threatened species, and now these are just skeletons. These are not living examples, but we can show you some living examples of these a little bit later. Any one of these, you can pick up, have a close look at. Some of the species that are closely related to the Acroporas are things like the Montiparas and the Pavonas. These species are typically more autotrophic. Do you know what I mean when I say that?
I learned that somewhere. It makes their own food.
Sort of. So how might a coral make its own food?
Photosynthesis, I would assume.
Sure. So because they have algae that live in their tissues, they are able to get their own energy. Most of these are all in the genus Favia or Fabites. These types of corals, they grow as boulders. Their tissue is very slimy. They create a lot of mucus because they're more heterotrophic. So that's to say that the coral animal does most of the energy acquisition by eating. The reason they're slimy, or the reason they're covered with mucus, is to help with trapping food particles. I'm not sure how familiar you are with this project. It's been quite popular, but these tiles, they're printed out of terracotta. So there are a few places in Hong Kong now that can do this, but it's essentially a robotic arm that is able to, we can control it with a computer to print these different shapes. And we tried to make shapes that kind of look like the surface of a coral, and that's just for aesthetic purposes. But every coral that you see in this tank is native to Hong Kong. You can see that these corals are very, very brightly green. It almost looks like they're fluorescing. And it's because they're growing under this purple light that has those very high intensity wavelengths that's very good for photosynthesis, but it's causing the corals to express those genes that produce the enzymes that act as a sunscreen to the coral.
What kind of light is this?
I would have to look at our app to know the exact wavelengths. Corals, But there are some species in here that aren't doing so well. For example, on the far corner over there is a zoanthid. It's closely related to a coral, but it's actually a different kind of organism. It doesn't create its own calcium carbonate skeleton like a coral does. It is also expressing those very bright colors, but most of it is actually dying back because there's not enough food in here. Hong Kong corals have acclimated to an environment that's high in nutrients, so there's a lot of food in the water. And so it's sometimes very difficult to keep local species of corals in aquarium settings like this. If we use just boulders, we find that the coral doesn't grow very well on boulders. But terracotta, it's inert. We're not introducing any nasty chemicals. It's essentially just, yeah, it's mud, essentially. It's basically just mud that's been fired in an oven. And that's why it's hard. But in addition to that, though, because it's made of mud, after a number of years of erosion and breakdown, these will disappear back into the sediments. Not before you establish coral colonies on top that no longer need that structure. So the idea is we put the structure out there, we attach some small fragments, those fragments grow over the years, and once they're big enough, the clay will have dissolved. So it's like we haven't even introduced any kind of human footprint to the environment, which is, it's really nice when we can do it. We put those in aquariums to agitate the surface of the water, and yes, to create current.
Do you have to feed that?
In here, yes. We use a number of different things. We feed the corals with artemia, which is they're kind of like, have you ever had sea monkeys? Have you ever kept like
Sea monkeys?
Are they
Yeah.
like crabs?
They're
Shrimp.
yeah, they're shrimp. Very, very tiny shrimp.
Alright.
Sometimes, if you're like a little kid and you want to keep a pet, but your parent doesn't want to buy you a fish or something, you can keep a jar of sea monkeys. They're really just tiny. But corals eat them. Some of the fish in here eat them. We try to keep other organisms in here, such as the snails. There were some sea cucumber in here. I don't know if there still are, but it is useful to have some herbivores in a tank like this. You can see there's a lot of algae, and a lot of this is actually all cyanobacteria that's growing on the sand.
The brown stuff?
Yes, and that's just that also gets a lot of its energy from sunlight, and so we don't necessarily want that in the tank. So we try to keep these tanks, other organisms in here that can keep that back a little bit. A company called Reefroids that we use, and that adds a little bit of calcium to the water. It adds a little bit of nutrients to the water that it helps corals build their skeleton, and it also it stimulates their growth hormones as well, allows them to grow a little bit. We're not going to handle any of the live corals in this tank because, again, these are in a much more sensitive kind of setting with the lights and being in an enclosed system. But I want to take all of you outdoors to our touch tank where we can actually see some corals. Yeah.
I'm sorry.
Are these dying corals that need to be
So this is really just for display. This tank, we keep it here just to anyone who visits SWIMS. We do a lot more workshops besides just teaching about corals, but we still think it's important to be able to showcase to people that don't necessarily go in the water a lot what a natural, pristine environment looks like in Hong Kong. You can see an environment like this if you jump in the water around Sai Kung, or if you take a ferry boat up to Tung Ping Chau. This is what it looks like underwater. If the water's a little bit more clear, there are a plethora of coral species. They might not be so brightly colored as this, but they are as diverse as this in shape and in their genetics. So that's why we have these corals in this tank. They're basically the remnants of various experiments and other work that we've done. All the skeletons that are upstairs, those are corals that were used in experiments, and maybe we pushed them past their, their heat tolerance. But they've all been, and we've tried to preserve the skeletons as best we can as teaching tools. And it's all about trying to influence the people of Hong Kong to appreciate the biodiversity that's here.
Do we need to change the frequency of light in different circumstances?
Well, these lights happen. We have these lights come with like a phone app, and that allows us to set like a time to it. So you wouldn't leave these on 24/7. They do need a night cycle. And so if you buy Aquarium, there's so many different companies that make aquarium lighting, and there's so many of them now you can control with a phone app. If you look up like Reef Aquariums AI, you'll find this product. This isn't the only product out there. There are so many different things to choose from. We use these because they have a really great
Like you tend to make one by themselves with more reasonable price that they can afford.
Sure.
Yeah.
I'm pretty sure these are actually kind of expensive in my opinion. But,
That's good. That's good.
But they do have a really great app that allows you to really control all the different light settings.
Is it like to the like numbers using numbers?
You can set it to the nanometer of a spectrum that you want. You can also set like your day-night schedule, and there's a feature where if you say, okay, I want my day cycle to start at 7 a.m., it won't just come on. It'll have like a sunrise effect. Wow. So the light will kind of gradually come on, and that's all automatic. You don't have to go in and turn anything on. You can actually watch it happen in real time on your phone as the light goes up. You can watch it happen on a little thing. It's they're pretty cool. But like I said, we're not sponsored or anything. Water is constantly flowing through these systems where it doesn't actually matter too much. You could say the same thing for a shallow coral community out in the ocean. How do they acclimate when it rains? And the answer to that is they do have biological responses for when the water becomes a little bit less salty when it rains. But just to give you guys an example of what some of these corals look like in a more natural environment.
Oh, is this soft?
You guys should touch.
On top?
Yep. So this coral is called Goniopora, and you'll find that after you kind of agitate it a little bit like that, the polyps.
It goes in.
That's right. They retreat back into their corallites. So that's this is a response to being kind of agitated a little bit. This coral, you saw them kind of all out and extended. This is a coral that is very, very autotrophic. Now, the reason they extend their polyps like that, you might guess that it's autotrophic, being it almost looks like a ball of anemone, but it's really because they're extending those polyps to increase their surface area. So it's not that much of a slimy species either. There's not a lot of mucus on the outside. Contrast with a brain coral like this one, if you touch that, you'll find there's quite a lot of mucus.
Oh, slippery.
Yeah. It's a little bit slimy.
Slime.
Yeah. Now, as someone who teaches diving and hosts snorkeling trips a lot, I must emphasize that when you're out there in a natural environment, it's never a good idea to touch the corals or any other live organisms. This is a very controlled situation, and I keep this tank the way I do specifically to give people that hands-on experience to appreciate these animals and Hong Kong's natural wildlife. That's why we have this tank. So it's okay to do it here, but I just always emphasize to everybody when we're out in the ocean, we never do that with live corals. Now this is a piece of Solitariensis, the plating coral that we saw the skeleton of. You'll notice, we can take this one out of the water. You'll notice that part of this coral is not doing so well. It is a more sensitive species. These dark browns, that's the living part of the coral, but if you touch that, you'll find it doesn't feel much different than the skeleton that you would have held on to upstairs. Just to emphasize that point, that the animal itself is just that. Yeah.
Is the white part like bleaching?
On that particular piece, it's simply dead. It hasn't been dead for long, otherwise it would be covered in algae. So like if you compare the different parts of this piece, this right here has been dead a while. This is more newly dead. I don't know if I have an example. Oh, right here. There's another little branching coral here that is bleached. So this is still living coral tissue. You can see there are patches of some of the polyps that still have algae there. That's why it's all like polka dot almost. That's still living tissue, but it's just a piece of bleached coral. You know, and there's a number of reasons for that. This is a fragment, so that process of fragmentation, which would have happened recently with this particular piece, is what it could be, that stress response. You know, it's always hard to tell. Let's see. So inside there was a coral, it was like a leafy coral that was bleached that I pointed out. This is what a healthy coral of that same species looks like. There's like, you know, the polyps, the tentacles on it, you can see are kind of green with that brown backdrop of the rest of the flesh of the actual coral. There's a lot of this particular fragment this is all just dead skeleton here, but you can kind of see where the tissue ends and the skeleton is just kind of dead. You see the difference? It's where all those little green tentacles are. And the reason this one's much more healthy is because being an autotrophic coral exposed to a natural day cycle of natural sunlight, it really is the best kind of situation you can put at least a Hong Kong coral in, with access to seawater. You know, there's tons of micronutrients in here. There's microscopic algae and zooplankton that some of these corals are eating. That's really important, especially when we're studying something about the natural system of the colony, the natural biology of the coral itself.
Like a plant would do better outside.
Exactly, yeah. Obviously, there are certain types of plants that do better inside, but with Hong Kong corals, they're going to be doing much better outside. We even have some fish in here. I don't know if you can see some of the things moving around.
I saw crabs.
There's lots of stuff in here. There's hermit crabs, snails.
Better.
Do any of you guys know what that is? You can hold these if you want to.
Yeah.
Whoa.
They would sometimes go.
You want to touch it? He wants to.
Feels so good.
Here, you can. I won't do that again.
Interesting.
Yeah. So sea cucumbers. So this species, if you go to the dry seafood markets, you'll see them. They look like little shriveled up prunes. Yeah.
A Chinese name.
Even though, you know, this species can get to be the size of my arm, but they'll still dry up to be those little tiny little prunes because it's mostly water. Their whole ecology is just to move around, you know, the bottom. They're taking up all the bits of sand and rocks, And inside their stomachs, they just dissolve and digest all of the organic matter that's covered in all of that bottom material, and then they just, you know, poop out those rocks again, all clean and nice. So they're like our vacuum cleaners. Yes, and it's one of the nice reasons why I have them in here. It's to keep the tank environment very healthy.
Does it have any relationship in the ecosystem with coral reef?
Yes, anywhere where you find corals, you'll find sea cucumbers. It's part of the ecology of these systems. Coral reefs, I'm sure you know, they are the most biodiverse marine ecosystems we have. Without the corals, you don't have a habitat for all of the snails and hermit crabs and cucumbers and fish and urchins. Without all of those other organisms, the coral would die because everything has some kind of ecosystem function. Everybody plays a role out there. And sometimes that even means cleaning the surface of the corals. If you look at some of the brain corals, they'll have snails on them. They'll have shrimps on them. Things that remove parasites and sediments. Everything is working together in a ecosystem, and that's really what this tank is meant to represent. It is an introduction into a complete community environment that, and everything in here, by the way, is from the Marine Reserve. So the body of water just in front of this building is Hong Kong's only marine protected area. We do workshops with students here very frequently where we take just one sample about the size of a basketball. And sometimes there's so many different species on our sample that we use that to occupy this tank. The only things that don't come into this tank on that sample are the corals. The corals are specimens that have been, just like the tank indoors, they've been part of different types of projects over the years. We never let them grow super large. We fragment them and we attach them to small pieces. This is our way of coral farming. We actually farm corals. We take large colonies that don't grow very quickly. We chop them into smaller pieces. What that does is it brings about a growth response, and so small pieces grow quicker. Whenever a coral gets so big and we don't have any room to do any more fragments, we return that back to the environment. So we've had such a little impact. So it's a process we call coral farming, and it's a really positive coral restoration technique. Just like the sea cucumbers, they use their mouth to just eat stuff off the bottom.
Oh, it's moving.
Of course. It's a live animal. Everything in here is alive and doing some kind of interesting job. You can see a really interesting fish along the bottom there. If you guys have...
Well, it's looking up to it.
You can hold it if you want to. Suction cups and tubed feet that allow them to move around, and that's how they practice their locomotion. If you want to hold it, so if you want to have it walk around on your hand like that, you have to get your hand wet for that to happen. Just scoop up a scoop of water in your hand. Yeah. Get your hand wet. Just keep your hand flat.
Whoa. Why this one looks so different than that one?
It's a great question. So you've identified, this is actually not a coral. This is another...
Oh, is it the one that's inside?
Yes.
The green one.
It's a different species to that one, but it's the same kind of animal. Touch that.
Immediately
Yeah.
scrunched up. Oh. It was so mucusy. There's so much mucus.
This is called a zoanthid. They're like... I don't even know if there's a common name for these, but very, very common in the, if you go to the goldfish markets, you'll see they will actually, you have to be careful because they actually sell these as corals, and they are not corals. They're closely related to corals. They have the same symbiotic relationship with algae, and so, it's important to keep them in a lot of sunlight, but they're not actually corals, they're zoanthids.
How do they differ?
So, zoanthids don't actually secrete calcium carbonate. This is actually just growing on a rock. So they'll cover a surface. They're more like a colonial anemone than anything else. The corals have evolved that ability to create the hard calcium carbonate skeleton. The zoanthids don't do that. They're in the same phylum. Do you know what phylum? You know, I bet you've heard of it before.
Yeah, it's familiar, but I don't remember.
The Cnidarians?
Is it like a group of a family?
Yes, it's a, so you have these different biological terms. You have the phylum, so that would be things like corals, zoanthids, jellyfish, sea anemones. These are all cnidarians. Do you know what a nematocyst is? So all of these organisms are all stinging organisms. Their cells, the outer layer of their tissue, has stinging cells. In jellyfish, it's much more pronounced. That's why if you get some jellyfish tentacles on you, sometimes it hurts. A nematocyst is a microscopic structure. It's like a little harpoon that will fire when it's agitated, and you or I might experience it as a little sting. But the way a jellyfish eats, you know, it's just in the water. It's basically just stunning any kind of shrimp or small fish. Some organisms have become immune to this ability. So like clownfish that will live in anemones. There's that scene in Nemo.
Where
they feel like they have to brush. Clownfish will actually do that as a way to keep parts of the anemone from being stinging so that they can safely live in the anemone. And it's more like something called a commensalism, where one organism, the clownfish, benefits from that habitat. And,
does the anemone get anything from the clownfish?
I believe some anemones that have clownfish will benefit from the clownfish eating parasites off of the anemone. But anyway, corals have this ability as well. Corals have evolved to utilize sunlight more so than some of those other organisms, but that's just one unique feature about corals. But these are all the phylum. When we get down lower to like orders and families and classes, this is how we differentiate some of the different groups of corals apart. So it's that biological hierarchy of organisms.
The phylum's really high, right? Really big. Right?
Yes, exactly. Above phylum is kingdom, which would be the animal kingdom. So, you know, you and I, we are in the same kingdom as pretty much everything in this tank, but we're in a different phylum. We're in the same phylum as the fish. We're also in the same phylum as the tunicates, which is a surprise to most people. Out of all of the invertebrates in this tank, this is our most common shared ancestor. These little sea squirts.
Oh! What was that?
They're
Is it alive?
Yep, those are solitary ascidians. They're and the way we know that they are in our same phylum, when those are developing, they actually have something called a notochord. In big vertebrate mammals like us, our notochord, when we're just an embryo, that ends up becoming our spinal column. In these organisms, it's really kind of a reduced structure, but it's present in their development.
So it has a spine.
No, it does not have a spine. It has a developmental structure that has a similar developmental stage as this. But in terms of an evolutionary history, like how closely things are related, it is closer to us than any of the other organisms in this tank. That is actually a bit unrelated. So when a coral fluoresces, like you saw in the tank inside, that is a response by the coral. When a coral is bleaching, one of two things is happening. Either the algae vacate, or the coral kicks them out. And the algae might leave because the coral is very stressed. So bleaching is a stress response. The tissue fluorescence is a different stress response. When a coral fluoresces, that's a stress response from too much light. Bleaching is a stress response usually because the temperature of the water is too high.
But what if I just do the light for like 30 seconds, just give enough to those polar bears?
In my opinion, 30 seconds of really intense light is probably not going to give you much of a response. I mean, bleaching and creating so those enzymes are really a secondary metabolite of the coral as a response to being in an environment that is very, very well lit all the time. If you're thinking about trying to bring about that effect in corals, you would really have to keep a coral in a high light environment for a long period of time. You won't necessarily see a coral fluoresce bright green like that if you just flash it with 30 seconds or a period of light. It has to be existing in that kind of environment for an extended period of time.
The book fluorescence is not really unhealthy for the coral. It's just a response to the fact.
It's a little bit of a nuanced question because sometimes if you think about some of the species in this tank and the environment that they exist in an environment that has a lot of nutrients which cause the water to be filled with algae. It's difficult to see that in a tank like this because we're getting flushed out with lots of clean seawater. But if we went snorkeling right now, maybe if you're standing where you are and I'm standing where I am, I might not be able to see you right now because the water has so much algae in it. And because of that, the corals are not being hit by such an intense amount of light. If you take a coral like that and you put it in an aquarium and you shine that much light on it, you will get that fluorescent response, but you also might be triggering other stress responses that might cause bleaching at the same time.
Bleaching is essentially a stress response. It's a way to preserve nutrients or energy to itself, to like give up on part of it.
Well, sunlight can cause all sorts of damage if there's too much of it. And the enzyme that a coral produces, which causes it to fluoresce, also happens to protect its tissue. So when I say that they create that, it's something that we call an epigenetic response, where a part of their genome is activated when they're in a situation where there's so much light to create that enzyme that you and I see as a fluorescence to its tissue. Well, the reason they're doing it is because that enzyme happens to protect the tissue against too much light. So it's a stress response in the sense that I'm stressed, I'm producing this enzyme to deal with that stress. We call that a stress response. Does that make sense?
So it's like trying to protect itself.
Exactly.
I bring this slide to them and I just want them to produce those slides for one time. Is it okay or is it not really healthy? Just one time to find out if there's any coral reef in their area.
So basically we're checking on, like, we're trying to detect a spot where there's coral and automatically. That's like a part of our project. And
Okay.
she's trying to ask is whether we can shine that light in the ocean to basically spot the coral.
I don't think that's going to harm coral, if that's what your question is.
That's mostly your question. But would it work in murky water, as you have said? The water's not really that visible.
Well, like I said, you're only going to get that response when you expose a coral to an extended period of it. So if you were to just go snorkeling with like a really bright fluorescent light and you shine it on some corals, that's not going to harm the corals at all. They're not going to die. They're not going to die. They're not going to experience that as a stress unless you're, unless you're sitting there underwater on a snorkel for many hours at a time, just pointing a bright light at one coral. Maybe that would do something, but I don't think that's what you're planning on doing. Maybe I'm wrong.
It's not gonna work. Because we're trying to like build like a drone and to scan the ocean, and for it to make coral more visible. She did some research and found out they fluoresce. So she's asking whether shining a light would work, but you need to shine it for a long period of time, so it probably wouldn't work.
Well, I mean, what you can do, like I think now I have a better understanding of what we're trying to do. There is like there are ways of being able to detect photosynthetic organisms using, this is a bit different than when I've been describing the bright corals that we saw inside, I've been using the word fluorescent. Photosynthetic fluorescence is something that you can detect, but it's very different than what the coral is doing in the tank. I'll show you guys something upstairs. All right, I'm going to test your knowledge a little bit. So, do you guys know what makes land plants green?
Chlorophyll.
Chlorophyll. What cell?
Chloroplast.
Yes. The chloroplast is the organelle inside the mesoderm of the plant cell. The chloroplast is filled with chlorophyll pigments, and that's what makes most plants green. If you think about what's happening within the chloroplast, there are stacks of grana, also called thylakoids. I don't know if you guys remember any of this from, it's like plant cellular biology. But that's where photosynthesis happens. It's within the membrane of those stacks of grana. On the surface of that cell, there will be there's that lipid membrane, and within the membrane of the thylakoid, you have all these different cellular structures that when photons are hitting the surface of this leaf, when light is hitting the surface of that leaf, they bring one photon will have one electron attached to it. And what's happening many millions of times within 1 millisecond, photons are hitting the surface of these leaves, and the energy, the electrons from those photons are being transferred from that very cellular surface of the membrane to the different chemical reactions that create the sugars of photosynthesis. It's a complicated biochemical process, but the reason I kind of bring all that up, while photosynthesis is a very efficient biochemical reaction, it doesn't utilize all of the light. So right now, we don't have a light turned on in here, it's a bit shaded. After a short period of this, I just got this out of a lit window. After a short period of time, the rate at which photons can be accepted by the photosystem will actually increase, because now that it's in the shade, the cellular mechanisms are like, where's all the light gone? I'm ready for more, I can do more. But if the plant is in the window, it can be saturated with light. It can be, I can't actually do anything with all of these electrons. There's a few things that can happen with that surplus of light, of those electrons. They can be given off by the plant as heat. Not heat that you or I can detect, but it's given off as an additional source of energy as heat from the plant. Not that you would ever feel the heat. It's such a tiny chemical amount of heat.
Electrons, right?
Yes. One other way that the plant might re-emit those photons is something called photofluorescence. And that's what this machine detects. So this machine is emitting a very small amount of blue light from this sensor here. I don't know if your camera is going to pick up on it.
Yeah.
Okay, and you can see it flashing.
Yes.
This machine is designed to emit a little bit of light out from this detector, and then so this is a fiber optic cable. Any light that is fluoresced back from that photo system it passes through this fiber optic cable, and there's a detector inside this machine. So if I turn this back on, if I shine this at you'll see that this is dancing around because there's a light environment here where photons are jumping around, they're bouncing off things. If I shine this at my hand, that number is not going to do very much. It's going to stay below 100, this F value, okay?
Oh, is this fluoro?
It stands for fluorescence. If I point this at a leaf though, you'll see that number jump up. So in real time, we are measuring the plant fluorescence on this. We're able to measure that because the machine is sending out a specific amount of light, and it is detecting that light that's coming back at a different wavelength, that fluorescent light. So now I can't remember why I'm telling you all of this. The original question was to
Detect coral.
so the same way that we're doing this on this plant, you can do this with the surface of a coral. It works the exact same way.
So you is that it won't have, it will be different output from this machine reader.
It'll be very similar. That number, that F value, is going to be much higher on a leaf from a land plant than it will on the surface of a coral. There are lots of ways that you can change the settings of this machine to be specific for corals. By the way, this is also a diving pan. It's in a housing. This can all go underwater. So sometimes we actually take this deep underwater, and we can do this underwater. The way we might use a tool like this, sometimes if a coral is stressed, if we want to detect whether or not a coral might be stressed, we would grow a coral in a natural environment, take regular measurements with a tool like this. And then if we expose that coral to some kind of experimental setting, whether we increase the light, maybe we put it under a different kind of light, or we increase the temperature, we can take the same exact measurements in the same exact way, and if we detect that there's a difference in the fluorescence yield of that organism, we might say that that is a stress response from that organism. This is a very powerful tool that can measure all sorts of things. If, for example, we took this plant and we measured what its fluorescence was just in a lit window, let's say, we might get, it might not be a very meaningful measurement. High, low, we don't really know what that means until well, one thing that we might do, we might put it in a room and shut all the lights off. We might be simulating nighttime, essentially. And then, if we take our probe and we shine it on the plant, we might get its dark acclimated state. If you think of that thylakoid membrane again, in the dark, all of the photoreceptors of the photosystem are going to be ready to accept sunlight. That's just the normal state of a plant. It wants to make sugars for growth, for reproduction. And if it's nighttime, it's ready to receive sunlight. If it's daytime, it's not. It has a surplus of light. It's saturated. So if we take a plant and we keep it in a dark environment and we take a series of measurements, we might know the yield of fluorescence in a dark acclimated setting. And then if we slowly, gradually move it into a light environment, to simulate a sunrise, and we take the same kind of measurements, we can describe how that organism acclimates to a light environment. One thing we can do with this machine, turn it on again, I'm going to give it a saturation pulse. I'm going to give it a saturation pulse.
Oh.
It's a very bright amount of light. It's more light than this plant could ever possibly be able to use. And we know at that point, basically the maximum amount of light that it could use in both a dark setting, in a light setting. And that can tell us a little bit about the photosynthetic efficiency of that organism. Queensland. But this method for detecting coral bleaching has been adopted. Basically, print a large size of one of these color cards. You can cut out this middle bit so that you just have basically
Too much light damages or the coral or makes it unhealthy. Is there a time length that is best for the coral's growth and optimal time?
I'm sure the answer to that is yes, but it's going to be so different depending on the species of coral and the different community of algae that lives within the tissue of that coral. You know, so it's a very difficult question to generalize an answer to.
One last question.
Yeah.
Sorry. Do we currently have any technology to get this a search under the sea, see the area of coral reefs in Hong Kong or wherever?
Well, like how would you go about seeing?
So I mean, like for example, if you wanted to do research on that area of the sea, how can you make sure you have coral reef over there?
I mean,
swimming over the Hong Kong.
there's enough of a dive community in Hong Kong. If you go to YouTube, depending on if you know where you think you want to look, if you go on a YouTube and you look diving video, then just hit that site, you'll probably be able to find somebody who's done some video.
Okay. Because I keep asking questions during these 2 hours. I'm quite embarrassing, but yeah, thank you for your
Thank you.